Hello, I'm Dr. Afsar Ahmed from Latrobe University and Atana Health Services. Before I begin, I'd like to express my sincere gratitude to the editorial board for inviting me to contribute this review article and for providing me with this opportunity to share a brief summary about this work. Therefore, I'm quite pleased to present an outline of this review article entitled Targeting Integrin in Immunity, Precision Matters for Therapeutic Interventions. Integrins are transmembrane cell surface receptor that mediates communication between cells and their surrounding environment. By linking the extracellular signal to the intercellular signaling pathways, they regulate numerous biological functions, including cell migration, proliferation, differentiation, cell survival, as well as immune response. Because integrins are also implicated in numerous diseases, including cancer, inflammatory disease, and autoimmune disorders, they have long been considered as an attractive therapeutic targets. However, despite extensive research and drug development efforts over the last 40 years, only a limited number of integrin-targeted therapies have achieved sustained clinical success. In this review, I discuss one of the key reasons for these challenges. Integrins are not just a simple on and off switch. They comprise a diverse family of integrin receptor with complex activation pathway, overlapping functions, and highly context-dependent biological roles. Same integrins can be beneficial in one cellular context while contributing to disease in another. My earlier work, along with the emerging evidence, suggests a very essential role of integrin within our immunity, and playing a very essential role, including maintaining immune homeostasis, leukocyte recruitment, macrophage polarization, toll-like receptor signaling, as well as anti-cancer immunity. Therefore, it is not quite surprising that targeting or global inhibition of integrin may lead to adverse side effect. This review therefore highlights alternative strategies that may offer greater therapeutic precisions, which include indirect targeting of integrins through the downstream signaling molecules. As an example, my earlier work was focused on one of those downstream signaling molecules called integrin link kinase, as well as cell type specific approach that can selectively target integrin function within a defined cellular compartment. The growing understanding of the integrin biology has therefore shifted the field from broad inhibition towards precision targeting. Future success will likely depend on not simply blocking integrins, but on defining which particular functions of integrins driving the disease and how they can be selectively targeted. In light of this review, I would also like to emphasize that the broader complexity of the biological system should be carefully considered when designing the therapeutic strategies. Molecules are often targeted based on their disease, based on their role in a different kind of like a disease, but yet many of these molecules do play very essential physiological roles. A failed therapeutic outcome does not quite necessarily mean that target is wrong, but more often it suggests that we need a better understanding of how, where, and when this target should be modulated. Only by embracing the biological complexity and by developing more precise therapeutic strategy, we can improve our chances of translating significant biological discoveries into effective and safe therapeutic options. Thank you for your interest.